All right, we'll give everybody a couple seconds to trickle in here, and then we can get started. All right, hello everyone, and thank you for joining us for today's webinar brought to you by 2020 Seed Labs. My name is Quentin Moorhead, and I'm the National Account Manager of Top Crop Manager in Potatoes in Canada. I'm joined today by Trevor Bluab, Moses Palmer of 2020 Seed Labs, and Tom Jensen of Jensen Agro. Today, during our session, Trevor, Moses, and Tom will discuss seed health, encompassing germination, vigor, and disease screening. They'll also touch on soil health and the importance of understanding nutrient, location, and utilization. Thanks to 2020 Seed Labs for sponsoring our session today. This webinar is being recorded and will be made available to all attendees and registrants approximately 24 hours after our live broadcast. The session is scheduled to run for about 60 minutes. The guys will speak for about 45, and then following their presentation, we'll open the floor for questions. If you have any questions throughout the presentation, please type them into the questions tab found on your computer screen. This webinar has been approved for half a CCA CEU in crop management and half a CCA CEU in nutrient management, and further instructions for submission will follow the presentation. Without further ado, I'll let them take it away.
Good morning, everyone. We'll be looking at germination tests. What exactly goes into a germination test? What are we looking for when we run a germination test? I'll also talk a bit about vigour testing and why a vigour test is definitely different from a germination test. And then we'll round it off. We're talking about what are some of the things that we see in a vigour test and a germination test that could be really informative to growers in making decisions about seed treatment and so forth. In the critical test that we usually recommend for growers going into the season is a germination test targeted at identifying the genetic potential of your seed lot. And then we also talk about a vigour test looking at how your seed performs under worst case scenarios. And then there is disease screening, very critical. And then also 1,000 kernel weight test, and that gives you an idea about how much to seed and how that influences your plant stand. So why test your seed? I get this question quite a bit. And I guess for seed growers who are growing pedigree seed, the answer is simple. It's the law. You have to test your seed to be able to meet pedigree standard. But for an average grower who is growing and it's not necessarily a pedigree seed grower, we did some costing in our lab, and then we came to the conclusion that the cost of testing is the lowest cost inputs per acre when we consider all the cost that goes into producing 1 acre of seed. When you test your seed, you can determine that your seed is high quality. It leads to uniform emergence, uniform plant establishment. It helps the canopy to close earlier so that you have better control of weeds. And then it also helps you with crop staging timing of your application. Of your field are. A germination test, like I said earlier on, is a test where we're looking at the genetic potential of your seed lot. So we're asking ourselves a few questions in a germination test. We're asking ourselves the question one, if conditions are optimum, if we have enough moisture, if we have enough light, if the temperature is right, and we have enough free gas exchange around the seed, what is the potential of that seed to produce a healthy plant? When we talk about healthy plants right now, we are talking about the presence of essential structures that are healthy. So essential structures, I'm referring to your roots, I'm referring to shoots, I'm referring to cotyledons and endosperms, and I'm referring to that whole structure together. We have enough experience as agronomists and individuals joining this conversation to understand the influence or the impact roots and shoots and leaves have on plant growth and development. So I won't spend too much time going into that. A germination reports will normally present you with a percentage breakdown of the components of your seed lot. So the first one will tell you what percentage of your seeds are normal, and that is what we usually report as the germination percentage. You also have a percentage of abnormal seedlings. So these are seeds that grew in the germination test, but there were defects to essential structures to the extent that those plants would not be able to produce a healthy plant if those seedlings were growing out in the field. So those are the seedlings that we characterize as abnormals. And then there are seeds that simply do not grow in a germination test, and we categorize them into three main groups. The first one is dead seeds. So dead seeds usually are either seeds that have been injured in a way that prevents the seed from growing. It could be due to heating in the bin or heating during the drying process, or it could be that the seeds are just physiological dead because they have aged and they've reached points of physiological aging where they just can't grow. And so in a germination test, as much as we're looking to report the percentage of healthy seedlings, we're also looking for in the germination test, and we usually it's characterized by roots that do not grow down into the media. Simply trying to avoid the media. And this is usually as a result of the seeds coming into contact with a chemical agent. And it could be anything from a pre-harvest herbicide, or it could be some seeds that were stored in a container that had previously stored some herbicide or some fertilizer. And so we're able to diagnose that in a germination test, and we usually provide this as additional information on a test report to the customer. In years where we have a lot of dry conditions, especially prior to harvest in peas, we also see several signs of mechanical damage. And here I have healthy pea plants as we would normally see in a germination test, and I've contrasted that to seedlings that have mechanical damage. Now, the way the pea seeds are designed, they have two cotyledons on each side, and then there's the radicle and in the chat. Can everyone hear me? Okay. Hello.
Hey Moses, seems like you're having a
Yeah.
little bit of a connection issue here. So maybe we could flip over to Trevor's presentation and then we can come back
Okay.
to yours once your Wi-Fi stabilizes.
Sounds good.
Trevor, if you want to jump in and we can jump into your part of the presentation, that might work a little smoother.
Sure, sounds good, Clinton. Here we go. Okay, can you see it okay?
Betcha.
All right, I'll get started. Yeah, Moses is down in Chile right now, so maybe a little bit of unreliable internet, I think, at the time. So I'll be talking about pathogens, primarily seed-borne pathogens this morning. So I'll just get going. So I'll start out talking a little bit about plant pathology, just get everybody into that kind of mindset, thinking about diseases. I'll go over some protocols, how we test for seed-borne pathogens, what kind of tests there are out there, which ones to request. I'll talk a little bit about statistics, primarily with Fusarium graminearum, because that's the most testing that we do and the best data that we have. And then I'll briefly finish up talking about soil testing. So one of the things to always keep in mind is the disease triangle. So with the disease triangle, it's saying that the risk and severity of disease is really dictated by three factors. You've got your host, so this is your plants that are growing out in the field, whether or not they're resistant to certain pathogens. You've also got your pathogen. So this is going to be both like quantity of the pathogen that's present, but can be like the specific strains, whether or not it's virulent or avirulent. And then a big role as well as the environment. So most pathogens like warm and humid environments, but there are a range of different pathogens that like a range of environments as well. So with Fusarium graminearum specifically, we're talking about warm, specifically high levels of moisture during that flowering period. The other thing that we always keep in mind is life cycles. So plant diseases, they're cyclical around the crop season. One of the most important things to consider is how it's overwintering from crop to crop. And one of those ways is, of course, soil for soil-borne pathogens. A major way that they overwinter is on the stubble, and this is going to be anything that really affects the upper parts of the plant aside from the roots. That's going to be a major source of the inoculum. But of course, seed too, and that's going to be both these ones that affect the upper parts of the plant as well as the roots and things below the soil level. So really by following crop rotations, we're ensuring that the stubble gets broken down so that you don't have as high levels of specific pathogens in the field. Same thing with inoculum in the soil. If you're following your crop rotations with diverse different crops, you're going to be having introduction of different organisms in there, and you're going to have a quicker breakdown of pathogens in the soil. So it's important to follow crop rotations for that reason. When we're testing for seed-borne pathogens and soil-borne as well, we have overall two broad methods, each with their own pros and cons. So the first here is molecular testing. So this is basically we're just looking for the DNA of the pathogen. And this can be using conventional PCR, qPCR, ELISA, DNA sequencing. And the advantage is that this can be more sensitive. So we're able to test larger volumes of seed when we're testing seed and detect lower amounts of pathogen that's in there than we might if we were plating it out. It can also be more specific. So we can look for a specific race of a pathogen or a specific, whether or not it's virulent or not. We can determine that using DNA testing much easier than we can using traditional testing, which would involve, inoculating a plant out in the field to see if it causes disease. A big advantage with the seed is that we see a quick turnaround for the results. So normally a DNA test is going to be 24 to 48 hours, we say, and that's much faster than your traditional plate testing. On the downside, often, especially with our fusarium testing, it provides just a positive or negative result rather than a percent infected seed. So you just know it's there, it's not there. And quite often you need to know that percent infected seed so that you're able to make those management decisions. With traditional disease testing, this is what I do at the lab here. This is taking sterilized plant tissue, most of the time seed, and we place it onto a growth medium. And so with the fungal testing that we do, we use potato dextrose agar, which is just a broad spectrum fungal growth medium that most fungi grow well on. And we just let it grow and look for the different diseases. And one of the advantages is that we can test for a large number of fungi in a single test. So you can see some examples of some plates here. And you can see everything growing on there: Alternaria, Epicoccum, Fusarium species. And so that's one of the advantages, is that as long as we know it can grow on that medium, we can put it on and look for it. Whereas with DNA testing, we have to, use specific primers for specific pathogens that we want to test for. It does also give a percent infected seed result. So a normal test, we do 200 seeds. So we can say 5 of these seeds are infected, you've got 2.5% infection. And that could be too much for one grower, or maybe it's just fine to use for another grower and he wants to use a seed treatment. So it really helps with making those decisions. On the downside, it does depend on the skill of the analyst. It's not accredited testing. Most of the disease testing that we do, the one exception is true loose smut testing, and that's quite different. But there's no agencies out there that are sending out samples to different labs to make sure that we're doing the testing right. Contamination can be a big problem. This image here, this is rhizopus. This is a really fungal, or a really common fungus that's just out everywhere. So the spores are really easily airborne, and it grows really fast. So if you get, just a single spore landing on a petri dish, it takes over the plate in just a couple of days, and it outcompetes everything else that's on there. So we're always working hard to keep contamination down in the lab, and we've done a really good job. And we always have to maintain that, though, of course. It's also a longer wait for the results, usually 5 to 7 days, depending on the pathogen, whether or not it's fast growing. We just need that time for the pathogen to grow on the plates, right? So that we're able to identify it. And then, of course, some pathogens don't grow on artificial media. So, one common disease that we test for is clubroot in soil and on plant tissue as well. And that just doesn't grow on any artificial media. So we need to use a DNA test to confirm whether or not it's clubroot that's present. So I'll talk a little bit more about Fusarium graminearum. This, of course, causes Fusarium head blight in cereals. This is kind of, if somebody's going to be sending in a sample, this is the #1 disease that they're concerned with being seed-borne. Of course, it causes Fusarium damaged kernels, produces harmful mycotoxins, and it can be really bad in some years. This is the life cycle of it, of Fusarium graminearum specifically. There's a lot of Fusarium species out there. Fusarium graminearum is the worst for Fusarium head blight in cereals. There are other ones that cause Fusarium head blight, and we do see them, in seeds, but Fusarium graminearum is typically the most aggressive and produces the most mycotoxins. So that's why we're so concerned about it when we're doing the disease testing. So in this life cycle, it's not soil borne. It could be present in the soil. In that case, it's going to be causing root rots. But as far as Fusarium head blight goes, it's primarily going to be the inoculum's going to be from the crop residue, where you get these airborne spores that are produced that are able to go up and colonize the heads during the enthesis period, right? And that's really when the infection occurs, and you're going to be having higher levels of these airborne spores released right after rainfall. So high amounts of rainfall during that flowering period in Alberta here, it's like mid-July kind of. That's the highest risk for seeing lots of Fusarium graminearum showing up. And then after you get the heads colonized, you get Sporodokia produced up there, which is the asexual spores, and those get distributed down further in the plant into the stem, colonizes there. And then, when the seed is harvested, that stem goes onto the ground and forms perithecia for future years. Of course, the seed that's harvested also has Fusarium graminearum, so that can be a source of inoculum as well. Primarily, if you have high levels of Fusarium graminearum in your seed or other Fusarium species, this is going to be causing issues like seedling blight, root rots, crown rots that you can see here. And I mean, other Fusarium species, doesn't really matter the crop. There's going to be a Fusarium species that's causing some root rots, most likely. But the species will be different between different crop types. So that's why rotation, again, is really important. So when should you test your seed for disease? Right after harvest. Definitely recommend, as soon as you can, because gives you a good idea of what you're working with. Is it worth it for you to clean the seed? Should you be looking for a seed treatment? Do you need to just find new seed? Is it way too much? Because you don't want to be scrambling closer to when you're going to be seeding it, of course. And if you do see some disease present, you might want to be testing multiple bins or look at other seed options, because we do see variability between different bins, even from seed, harvested from a same field or same area. And then after cleaning the seed as well, before you're going to be putting it in the ground, it might change the level of infection, possibly. Definitely storage over the winter is possibly going to be changing what's present. In general, I'd say the level goes down, especially year after year. You're going to be seeing the level going down. Most pathogens don't survive well under the proper storage conditions, but there are some storage molds that can be a problem as well. So you want to be able to know if that's an issue for you before you're planting it in the ground, right? Knowing what's going in the ground allows you to make the best seeding rate decisions. If you need to increase that seeding rate due to some fusarium, you want to have lower tillers, so that you have more uniform emergence and more uniform plant stands, so that you can deal with disease later on. A little bit of the statistics here. So this is Fusarium graminearum in Alberta across all the crop types, and the percent that have tested positive. Generally, peaks are associated with wetter years. So going back to 2007, we barely had any. There was kind of an increase. Then 2011, it dipped down. 2012, we picked it back up again, and it went up. In 2013, 2014, similar to 2013, 2015, drier year, we see a big decrease again. 2016 was a very wet year, and we see a huge increase, over 20% of samples. So that's 1 in 5 were testing positive. That year, a lot of it was located in southern Alberta, but things have changed now. 2017, again, things were drier, big drop down. 2018, even bigger increase now, or decrease. 2019, it went just right back up to that same level, similar to 2016. We're seeing about 20%. 2020, we dipped down. 2021 was quite dry. It's down closer to 5%. 2022, we see an increase. And now 2023, we're seeing quite a high percentage testing positive. We're at 18.4 right now. And I'm always updating these. So the 2023 number keeps on changing as the year goes on. So we're almost at 1 in 5 samples testing positive. Where they're coming from is quite different. I'll get to the map shortly here. Looking at the crop types, we see it break down by the susceptibilities. So durum is very susceptible to Fusarium graminearum, and typically we see it being the highest. This year, because those durum growing areas were much drier, we see a big decrease in the durum. But where we're seeing that increase is the barley. Barley's at its highest level, highest percentage that have tested positive yet. So it's at 22.7% of samples that are testing positive for barley. And that's even higher than the wheat samples, which usually wheat is higher than barley. So, but we did see an increase in the wheat, just not as significant as barley. And then oats, we even have the highest percentage testing oats. Oats are quite resistant, but we've seen a few samples testing positive this year. Looking at the severity, so looking at a positive sample, what's the average percent that are testing positive? We see an increase as well this year from 2022. So the average positive sample is at about 2%. So that's like 1 in 50 seeds that has an infection. you think about it that way. And that's driven hugely by the barley again. So this year, when we're seeing a sample come in and we're seeing a lot of Fusarium graminearum on those plates, it's more than likely been barley this year. And in the past, we have seen quite high average percent infection in the barley, but it's been quite low for quite a number of years here, and so this is quite a significant increase. Both durum and wheat are actually down from last year. Looking at the distribution across the province, I just went to the past 5 years, so you get a sense here. So 2018 was kind of a drier year. 2019 here, we see along that eastern border with Saskatchewan, quite that's kind of the hot spot that year. 2020, it was dipped down. We're not seeing very much in southern Alberta. Again, kind of central Alberta is the big ones. We're seeing some up in the Peace Country as well. 2021 was quite dry year, even more so, and seeing the highest amounts are actually in southern Alberta that year. 2022, this was a little bit of an increase again. Same sort of thing where we're seeing east of Edmonton along that Saskatchewan border. Now this year we're seeing a little bit different situation where we're seeing to the north of Edmonton and west very high amounts. That's where we're seeing some of the highest percent infected samples coming from as well, and kind of going to the southeast of Edmonton. Southern Alberta was very dry this year, so they're not seeing a lot of Fusarium graminearum down there. And we picked up some up in Peace Country this year as well. Other provinces, they are usually much higher than Alberta, but again this year, both Saskatchewan and Manitoba had quite dry years, so we saw a big decrease in the amount of Fusarium graminearum from last year. The same thing with the average percent infection of positives. We're seeing lower levels than even Alberta, which it's not very common if you look at the history here. So that's it for Fusarium graminearum. I'll just briefly talk about the soil tests that we do for pathogens at 2020 Seed Labs. So we test for clubroot. So this is both soil and plant specimens. If you see some symptoms out in the field, you can send them in during the growing season, and we can confirm if it's clubroot or not. Aphanomyces euteiches, this is a big problem in peas and lentils, of course, over the last few years. Again, same thing, we can test both soil and plant specimens. And then soybean cyst nematode, which is kind of a growing concern in Manitoba. And if you're looking for some guidance in sampling and sending it into the lab, you can go to our website at www.2020seedlabs.ca and click on submit a sample, and you can see these cards that give you some guidance in submitting a sample. Okay, and there will be time for questions after everybody else, but if there's nothing that I'm able to get to, here's my contact information, and you can contact me after. Thanks.
Awesome. Thanks for that, Trevor. We got one more presentation from Tom Jensen here of Jensen Agro, and then we can get to some questions here. So Tom, if you want to turn on your camera and share your presentation, we can get started on that.
Okay, is that showing up okay?
No, it's just showing the full screen right now, Tom.
What was it? That should be better.
Gotcha.
Okay, well, it's a pleasure to be here with you today. I'm a soil scientist and agronomist. I'm representing Down to Earth Labs today. And I teach at the University of Lethbridge Soil Science. But I'll go through my presentation, and as Trevor said, we'll have time for questions here at the end. So plant nutrients are very important. Any individual crop will use all of these plant nutrients when it grows. And some crops use more, some less of some of these. But the carbon, hydrogen, oxygen come from water and from the air. But the mineral nutrients uh, come from the soil, and they're primarily absorbed through the roots of the plants. So this is a useful chart from Adrian Johnston when he worked at Agriculture and Agri-Food Canada in Melfort. And it shows the nutrient uptake. The blue is biomass. And then each of these individual lines is a different nutrient. This is nitrogen, potassium, sulfur, and phosphorus down at the bottom. So plants use an awful lot of nitrogen and potassium and moderate amounts of phosphorus and sulfur. But all of them, day zero was when it was planted. For the first week, at least usually of a plant or a little bit more, the plant is actually living off the nutrients in the seed. And of course, one reason you can get your seed tested is to make sure you have good, healthy, viable seed. And that nutrient content within the seed itself is extremely important to get that crop off to a good start. Once it establishes its roots and its shoots and it starts photosynthesizing, then it'll start using nutrients from the soil. At first, the seedlings are small, but when they get to their bigger vegetative size, then they use a lot of nutrients quite quickly. We call this the exponential uptake of nutrients. Then as the plant starts to flower and set seed, nutrient uptake slows down. And at the end here is harvest. So the soil itself, the real critical part is after the little plant inside the seed has grown and used the nutrients within the seed, it needs to be able to access nutrients through its roots from the soil. Now, in any one year, the nutrients that come to a crop come from many different sources. You know, far too often we think about the fertilizer as the source of nutrients, but the reality is a lot of the nutrients come from soil organic matter, from the clay particles. The legume plants get a lot of their nitrogen from the bacteria living in their nodules. We get some nutrients coming down in the rainfall, especially little bits of nitrogen and sulfur. And if you're irrigating, you're actually adding some of the nutrients through your irrigation system. There's quite a bit of dissolved sulfur actually in some of our irrigation waters. But, you know, quite often we concentrate on the fertilizers that we add, but I just want you to realize the plants don't discriminate where the nutrients come from. Once it's in the soil from the various sources, that's when they're able to access those nutrients. So here's just a picture. This is an illustration of the clay particles. They're often layered. A lot of the nutrient cations will adsorb onto the surfaces of the clay. They can come off the clay particles into soil solution where the plant can take them up. The bottom picture is a picture of humus or organic matter in the soil. It's very complex, has lots of cavities and pores, and is able to store a lot of plant nutrients for plants. And that's why our topsoil and the humus or organic matter content is so important. There are other nutrients like phosphorus. When we add it as a fertilizer in the soil, it reacts with other elements in the soil and is often precipitated out as tiny little particles. Now they can redissolve and help supply the plant, but it's very much affected by pH. If we can maintain our soils from about above pH 5.5 up to about 7.3, it's when phosphorus is most available to the plants. So there's a lot of soil characteristics, pH among others, that affect the availability of some of these nutrients. Of course, we can get fertilizers from nutrients from our fertilizers. Nitrogen is the most used fertilizer, followed by phosphorus, and then potassium, sulfur, and many of the micronutrients. They're not, every plant needs all of these nutrients every year, but fortunately, most of our soils will supply a lot of these nutrients, especially the micronutrients, on a regular basis. So you can get fertilizers. We can use what we call chemical fertilizers or inorganic fertilizers, urea, monoammonium phosphate, potash. But those growers that are growing organic crops, there are nutrient sources they can use, alfalfa pellets, seaweed extracts, etc. There's I've worked quite a bit with organic growers, and they need to supplement nutrients to their soils in the same way that commercial farms supplement using chemical fertilizers. So getting down to what I want to talk about today, nutrient diagnostics using soil testing and plant sample analyses. These are tools that we use to help us know how the plants will do or are doing. And sometimes we can make corrections and improve conditions for the crops. It's important to realize that nutrients can move within the plants. For example, if a plant is low in phosphorus or nitrogen, it can actually remobilize nutrients from the older leaves and move it to the growing point to the plant to keep the plant going. And some of the nutrients are not mobile in the soil. For example, sulfur. Once sulfur is used in the older parts of the plants, it can't be moved to the growing point. And this is important because if you're looking at visual observed symptoms, you have to know what part of the plant to look at and what deficiency symptoms. So we look at terms defining deficiency symptoms. Chlorosis is yellowing. Necrosis is dead brown spots or tissues. We can have intervenal chlorosis, dead spots or necrotic spots. We can have chlorosis of the veins. We can have chlorosis and necrosis on the leaf margins. And we can have leaf tip burn or necrosis. So there's a lot of different deficiencies in. If you rely on these, you have to get very good at figuring out what they are and what nutrients it might be. Here's phosphorus, for example, just as an example. If it's very severe on cereal crops, you can actually get the older leaves will be a little bit yellow, and you can actually have purpling of the stems. However, quite often in our crops, this is a picture of a canola crop. The one on the right is phosphorus deficient. The one on the left has sufficient phosphorus. And these were planted the same day on the same field. And the only difference is phosphorus availability. Now, if you had the whole field, like the deficient side, you would just think, well, it's a cool, dry spring and the crops are slow. But it could be in reality a phosphorus deficiency. And just go down here. Here's potassium deficiency. When it's really severe, we see this leaf edge burning of the older leaves. Here's canola on the bottom right, alfalfa on the bottom left. So each plant has slightly different effects when there are deficiencies. And here's sulfur. It's not mobile in the plant, so it affects the younger leaves. Here's sulfur deficient alfalfa, sulfur deficient wheat. But canola looks a lot different. When it's young, you get a stunted plant with curled leaves. Later on, the plant is less thrifty, less flowers. So the stage of the plant is also important to understand. Now, the reality, I could show you pictures all day long of this is a deficiency of potassium in a certain crop and so forth. But the reality is, in a lot of our fields, we don't see the severe deficiencies every year, or we don't see them much at all. And instead, we use soil and plant analysis to determine whether or not we might have a deficiency or are low in availability of a nutrient. We use soil testing. We take samples. We ship them to a soil test lab. They use an extracting solution. They filter it. They analyze the solution, the filtrate, and they give us an interpretation, which nutrients are more available, less available, and whether or not it might be profitable to supplement with nutrients through fertilizers. We do the same with plants. We'll take plant samples. We analyze them. We take a sample, send it to a lab, have it analyzed. And this is the basic idea of plant analysis. If the plant shows up that it's low in availability in the plant tissues, we call it be deficient. We're not going to get optimum yield. There's usually a range of concentration that is ideal for the crop to do well. And that's what we hope we're in this range. And in some instances, we can add too much of a nutrient and it can actually start to become toxic and we start to lose yield. So this is just an example. It's important to realize that each nutrient is a little different, how it can move in the soil, how it can move in the plant. This is an example from Northeast Saskatchewan, Aaron Baldwin. The only difference on the two sides of this field, this had seed row starter phosphorus. They ran out of seed row fertilizer and planted the rest of the field. And it was a cool, dry spring. Phosphorus doesn't move very well. The field wasn't deficient in phosphorus. They had fall banded a pretty good rate, 30 pounds of P2O5. And the only difference was a little bit of starter phosphorus in the seed row. So for the less mobile nutrients like phosphorus and potassium, placing that fertilizer close to the germinating seed is important. So the important thing is if we can use soil and plant analysis to assist in making decisions of fertilizer recommendations, and the payoff comes, we can get higher yields, better profits, and keep our soils healthy. And we can have questions after. Those of you that, here's a QR code for all of the presentations today. And do you want me just to leave that on, Alex?
Yeah, if you could leave that up there, that Tom, that would work good. We'll let some questions trickle in here, but like he mentioned, here's your chance to collect those that half CCA in nutrient management and crop management. So if you open up your CCA app on your smartphones, click scan course code and hold it up to that QR code, you should be submitted automatically. If this doesn't work, please email your first name, last name, and CCA number to topcrop@annexbusinessmedia.com, and we'll get you submitted there. So we don't have many questions coming through here, but we do have one for sure, and then another question for Trevor. So Trevor, why was barley so much higher this past year for Fusarium? Do you have any insight into that?
Not too sure. I don't know. Like I said, it seems like a lot of those barley samples that are coming in that have high levels of infection. We have seen in the past, barley being quite high levels of infection when it's positive, but usually it's less likely to be positive, right? It's more resistant to getting that infection than wheat or certainly durum, right? That might have to do with the way it spreads through the field, the way it spreads through the individual head. it's able to infect, more seeds within a limited time period, kind of, right, during that flowering anthesis period. As to why there's a higher percentage that are testing positive this year, I'm not too sure. Just possibly, when barley was in flower versus when wheat was in flower during specific time periods in certain areas could be, but I'm not too sure. I don't have a good answer for that because typically it's more resistant to getting that infection than wheat is.
Oh, that works. And then just the last one was if you're willing to share the test results map with anybody, one person in particular is asking via email. So we can maybe throw your email back up there too. And if anybody wants any of those results, they could reach out directly to you.
Sure. Yeah, I did email Farah back, and so I'm in contact with her, and I'll send her the maps directly. But yeah, I can put up my email here too.
Awesome. And well, it looks like that's kind of all we got for questions. So again, if anybody has any questions following the presentation, just please
Oh,
reach out.
another
Moses.
question just came in, Quentin.
Okay, perfect. Uh, can you sample for all nutrients, and how accurate is nutrient testing in leaf samples?
Well, you can take plant samples and you can analyze for all the nutrients. And for most of them, we have a good database to know whether or not that crop at that stage has sufficient of the various nutrients. Some of the micronutrients we don't have really good data for all parts of Canada. However, a lot of them we don't see many nutrient deficiencies. For example, iron and nickel and so forth. It could only happen sort of spotty through the country. So we can analyze plant parts and know whether or not most of the nutrients are available in sufficient amounts or not. And it's good to talk to the lab and your agronomist ahead of time. If you're trying to correct a problem, it's better to sample your plants early in the growing season, not right at seedling because they're still living off the seed nutrients. But once they go into that vegetative stage, that's the time to sample, have it analyzed. If you're short in a nutrient, you have a chance to supplement that maybe with foliar applications in season and improve crop growth. I hope that answers it. So
That's perfect.
yeah.
Um, David reached out here. You'd like Trevor and Tom's emails. Yeah, so those will all be found in the follow-up email as well. All their contact information. We're going to be sent along with the instructions for the CCA credits there. So they'll all be found in the follow-up email. You should be getting that in about 24 hours. So unless anybody else has any more questions, we can wrap this up here. So thanks for tuning in today, everybody. Don't forget to visit topcropmanager.com/webinars to view all of our previously recorded webinars. Thanks again, guys. Sorry, Moses, about the internet connection there, but glad we could make it work on the fly here.